With that in mind, we actually run a trial at the federal facility, CReSA at Spain. It's a bio-level 3 facility where they are specialized in virus research, especially African swine fever virus. This is the result of this particular research. The goal of this research is really looking at the role of spray-dried porcine plasma on pigs contacted with pigs we artificially challenged with African swine fever virus, and to see how by feeding strategies impact those artificially infected pigs and also the contact pig. Do they behave, does the diet actually increase or reduces the transmission rate between pigs in the same pen? And those are the goals of this particular research. This is the timeline for this research. As I mentioned before, we have 2 dietary groups here. Group A contains 8% spray-dried porcine plasma. Group B is a control feed. We had 24 pigs in this research. 12 pigs were allotted into Group A in the room, and another 12 pig was allotted to Group B to another separate room. Again, this is the timeline. All pigs were brought into this facility 4 days before the trial starts, and to give them time to accommodate to this facility. On day 0, we select 2 pigs from each group. Randomly selected them, and then we inject the live virus, the African swine fever virus, to these 2 pigs. We call them Trojan pigs. And then we observe those 2 pigs for 2 days. After confirming they all got infected with African swine fever virus, we put them back into their original pens. Then we studied those 2 pigs and also the contact pigs by measuring their blood, nostril swab, rectal swab to see whether those pigs get infected or not. The contact pig get infected or not. Then on day 6, what we find is those 2 Trojan pigs from, actually 4 Trojan pigs from both groups all developed high fever. Under the lab protocol, once they reached 42 degrees, long and heavy fever, we have to euthanize them because of the lab procedure. So we euthanized all 4 pigs from the 2 groups on day 6. Then we followed all the contacted pigs in those particular groups all the way to day 18 to see how many pigs get infected and how fast they get infected. To our surprise, very surprising, none of the pigs from either group ever had developed African swine fever infections. So, because of that, we decided to extend this research a little longer by introducing additional Trojans, additional infected pigs. So in this case, on day 19, we brought in another 3 Trojan pigs. So we randomly selected 3 pigs among those remaining pigs in those 2 groups. From each group, 3 pigs. We randomly selected them, and then again we injected them with African swine fever virus with about 1,000 doses of those Georgian strain. And then we put them back into the pen immediately. And then in this case, we did not euthanize those pigs even though they developed high fever. So we left those pigs in the pen all the way until they naturally died. The idea is to increase the threat of the threat of the virus and increase the infections. And then we can learn does the diet impact the rate of transmission among the pigs. Again, for the second group, we followed them all the way to day 31. At day 31, that's when we have to stop the study, and we euthanize all the pigs, and we collect the tissue sample, blood, nostril samples, and rectal samples for analysis. This slide shows the temperature of the group A, of those pigs fed with 8% spray-dried porcine plasma. As you can see on the bottom, you can see those are the date of the timeline of the study, and then each line on the top tracks the temperature of particular pigs. As you can see on this top side, after we inject those 2 children pigs with African swine fever virus, they quickly develop high fever, and by day 6, we had to euthanize those 2 pigs. Then on the right side, you can see we have another second round of Trojans, which are those 3 pigs we injected with live virus. As you can see, they also developed some fevers, some high fever as well, which are shown in the red. One pig reached a really high fever early on, and the other 2 pigs reached a high fever, then seems to stay stabilized. The other color lines over here are those contact pigs. As you can see, those contact pigs, they didn't really develop any high fever until up to the end of the study. If we compare to the control group, as you can see here, the control group, again, similarly to the plasma group, all the challenged pig, they all developed high fever pretty quickly after injection. However, the contact pigs in the control group, those pigs seems to develop the high fever much quicker in the early days compared to the control group. This slide shows the average temperature of the pigs, of those contact pigs in those 2 dietary groups, by comparing those two groups' temperature. As you can see on the slides, the blue line represents the plasma group, and the yellow line represents the control group. As you can see, for most of the time, and the average temperature of the 2 groups are below 40 degrees, so they don't have any fever. However, at the end of the study, the control group does develop a high fever more quicker than the plasma group, which are suggesting that the control group are being infected quicker and developing the clinical sign much quicker compared to the plasma group. Now, we also, collected all the samples from blood and nasal and rectal swab, and also, tissue samples, and and then later on we analyzed those samples with PCR test and try to look at the DNAs or the genetic materials in those samples to see to see the viral load in those samples. And this slide really shows the children pigs 6 days post injection, where what is the viral load between groups. As you can see on the top is the plasma group, on the bottom is the control group, and again this is the PCR test, and where we tested the CT value of a specific sample, blood sample, nasal swab, or rectal swab. As we all know, the CT level, the lower the number is, the higher the DNA copy is. I actually made a conversion from the CT value to the DNA copies. As you can see, on the plasma group versus the control group, either in the blood or nasal swab or rectal swab, the plasma group pigs, even with this injected children pigs, the plasma group pig really had a lower DNA copies in the blood and nasal swab and rectal swab, and the fold differences between control group are quite significant, 44 times, almost 60 times or 8 times in the rectal swab. So, again, even in these children injected pigs, by feeding plasma in the diet, it potentially reduces the viral load in those artificially infected pigs. We also collected the tissue samples and the lymph node from all the pigs, and the contact pig and also the challenged pig as well. This slide shows the viral load in different tissues of the contact pig at the end of the study by different dietary type, plasma group and the control group. As you can see, the blue one represent the plasma group and the yellow one represent the control group. A plasma group across the board in all the lymph nodes had a higher CT value, meaning they had a lower DNA copies, African swine fever virus DNA copies in all those tissues. So again, this is really suggesting by feeding a different diet, by feeding plasma, it actually significantly reduced the viral load of those contact pigs. So, in conclusion, we see a dietary strategy including inclusion of spray-dried porcine plasma really significantly reduced the African swine fever virus load of pigs in those tissue, in those lymph nodes. Also, it reduced the virus load in the blood and nasal swab and rectal swab as well. And also we see by feeding this spray-dried plasma, it can delay the infection on the onset of these clinical symptoms or in the contact pigs. Really, this study really scored our concept about the dietary role in helping pigs helping pigs not get infected or reduce the risk of infectivity.